Hey, I'm Noggs, and this is Noggs House. Today we're going to talk about primordial soup. We're diving into one of the biggest and most amazing questions in all of science. How did life begin? Think about it. Earth started as a lifeless ball of rock and now is covered with living things. So how did non-living chemicals transform into living organisms? One of the most famous ideas about this is called the primordial soup theory. Primordial means from the very beginning, so primordial soup is the idea that early Earth's oceans were like a giant bowl of chemical soup where simple chemicals gradually came together to form more complex molecules and eventually life itself. This hypothesis, remember that's a scientific explanation based on evidence, suggests there was a gradual transition from simple inorganic chemistry, that means chemistry of non-living things like rocks and minerals, to complex biological systems in Earth's early oceans, probably somewhere between 3.7 and 4 billion years ago. Today we're going to explore the foundational ideas behind primordial soup theory, look at the amazing experiments that support it, and also discuss some of the big challenges and questions that scientists are still working on. The origin of life is one of science's greatest mysteries, and we're going to dig right into it. The primordial soup idea was first proposed independently by two scientists, Alexander Oparin, a Russian scientist in the 1920s, and J.B.S. Haldane, a British scientist around the same time. So we call it the Oparin-Haldane hypothesis. They suggested that life arose from organic molecules that gradually accumulated in Earth's early oceans. Organic molecules are molecules containing carbon. They're called organic because they're associated with living things, but they can form without life too. So what was early Earth like when this was supposedly happening between 3.7 and 4 billion years ago? The atmosphere was very different from today. It was a reducing atmosphere, which means it had very little oxygen, almost none. Instead, it was rich in methane, ammonia, hydrogen gas, and water vapor. This lack of oxygen was actually important because oxygen tends to break down organic molecules. It would have made it harder for complex chemistry to happen. Earth also had powerful energy sources available. Intense volcanic activity spewing out hot gases, frequent lightning strikes providing electrical energy, and unfiltered UV radiation from the sun. There was no ozone layer to block it yet. All these energy sources could drive chemical reactions, breaking apart simple molecules and allowing them to recombine in new ways. These were the conditions that catalyzed, that means sped up or triggered, the initial chemical reactions that might have led to life. With all that energy available, something amazing could happen. Prebiotic synthesis. Prebiotic means before life, and synthesis means creating or building something. So prebiotic synthesis is the creation of complex molecules before life existed. The energy from lightning, UV radiation, and volcanic heat drove the abiotic synthesis of simple organic molecules. Abiotic means without life. These molecules form naturally through chemistry, not from living things. The molecules that formed are called monomers. These are small, simple molecules that can link together to make bigger molecules. Think of them like Lego blocks, individual small pieces. The key monomers that formed were the building blocks of life. These included amino acids, which link together to make proteins. Proteins do most of the work in living cells. And nucleotides, which link together to make DNA and RNA. These store genetic information. The carbon for these molecules came from simple starting materials that were around on early Earth. Formaldehyde, hydrogen cyanide, and formamide. Through chemical reactions powered by lightning and UV light, these simple molecules could combine and rearrange to create the more complex monomers. It's like the universe was doing chemistry experiments all on its own. The primordial soup idea was just a hypothesis until 1953 when two scientists, Stanley Miller, a graduate student, and Harold Urey, his professor, did an amazing experiment to test it. They built an apparatus to simulate early Earth conditions in the laboratory. They put a mixture of gases in a sealed flask, methane, ammonia, hydrogen, and water vapor, the gases they thought were in early Earth's atmosphere. Then they added energy by zapping the mixture with electric sparks to simulate lightning. They let this run for a week to see what would happen. The results were stunning. After just 1 week, the previously clear water had turned brownish red, and when they analyzed it, they found it contained various amino acids, the building blocks of proteins. They had created organic molecules from inorganic starting materials just by simulating early Earth. Later scientists repeated and improved the experiment, and they were able to produce even more complex molecules, including some of the building blocks of DNA and RNA. This provided compelling evidence, really strong proof, that life's basic molecules could form spontaneously on their own under early Earth conditions. The Miller-Urey experiment is one of the most famous experiments in the history of biology. So these organic molecules were forming, but where did they go? This is where Earth's early oceans come in. The oceans played several crucial roles. First, they acted as vast solvents. A solvent is a liquid that dissolves other substances, like how water dissolves sugar. As organic monomers formed in the atmosphere or on land, rain would wash them into the oceans where they would dissolve and accumulate. Over millions of years, the oceans would become enriched with organic molecules, creating the actual primordial soup, a dilute mixture of all kinds of organic compounds floating in the water. Second, the ocean served as dynamic reaction hubs where these molecules could bump into each other and potentially react to form more complex molecules. But not all parts of the ocean were equally important. Scientists think certain localized environments were especially good for driving chemical reactions. These include tide pools, shallow pools at the ocean's edge that get concentrated when water evaporates and receive lots of UV energy from sunlight, hot springs, where geothermal heat and chemical energy from minerals would drive reactions, and deep sea hydrothermal vents, underwater hot springs on the ocean floor that provide continuous energy and have mineral surfaces that could help reactions along. These special environments would concentrate reactants, the starting chemicals, increasing the probability that they'd bump into each other and react. It's like how you're more likely to meet someone in a crowded room than in an empty field. Having individual building blocks, monomers, is great, but life needs bigger and more complex molecules. The next critical step was linking monomers together into larger molecules called polymers. Poly means many, so polymers are molecules made of many monomers linked together in a chain, like linking Lego blocks together to build something bigger. Some examples: amino acids can link together to form polypeptides, which are the precursors or early versions of proteins. Nucleotides can link together to form nucleic acids, like RNA and DNA. But here's a big problem. Making these links require dehydration reactions. Dehydration means removing water. When two monomers link together, a water molecule is released. The problem is, this is really hard to do in water. It's like trying to dry off while you're still in the swimming pool. When you're surrounded by water, the reaction wants to go backward. The water wants to break the links apart rather than form them. So how could polymers form in Earth's watery oceans? Scientists have several ideas. Maybe the cycles of drying and heating helped. For example, in tide pools that periodically dry out in the sun, monomers would get concentrated and heated, allowing them to link together. Or maybe mineral surfaces, like clay particles, provided special spots where monomers could line up and link together more easily. This is still an area of active research. Now we hit a really tricky problem that scientists call the chicken and egg problem of early life. Modern life needs two types of molecules working together: genetic material, DNA, that stores information about how to build things, and enzymes, proteins that actually do the building. But here's the problem. You need DNA to tell you how to make proteins, but you need proteins to copy DNA. So which came first? It seems impossible. The RNA world hypothesis offers a brilliant solution. RNA is a molecule that's similar to DNA. It can store genetic material just like DNA does. But here's the amazing thing. RNA can also act as an enzyme. Some RNA molecules called ribosomes, RNA enzymes, can speed up chemical reactions, including the reaction to copy themselves. So RNA has a dual role. It can both store genetic material and perform catalytic functions, act like an enzyme. This means RNA could potentially self-replicate, making copies of itself without needing DNA or proteins. The RNA world hypothesis proposes that the very first form of life was based on RNA, not DNA, and proteins. RNA molecules would have stored information and also done all the chemical work. Only later in evolution did DNA, better for long-term storage, and proteins, better enzymes, take over most of these roles. It's an elegant solution that bridges information storage and function. Even if you have RNA that can self-replicate and act as enzymes, you still don't quite have life. Modern cells have a membrane, a boundary that separates inside from outside. This is crucial. Without compartmentalization, dividing space into separate compartments, all your precious molecules would just diffuse away into the ocean. You need some kind of container. This is where protocells come in. Protocells are primitive-like compartments, not quite living cells, but simple bubble-like structures that could have been the precursors to real cells. The amazing thing is that protocells can spontaneously form. They assemble themselves. Certain molecules, like fatty acids, simply oily molecules, naturally form bubbles in water, with the oily parts facing inward and the water-loving parts facing outwards, creating a membrane. These bubbles can trap other molecules inside. The importance of compartmentalization can't be overstated. It concentrated reactants inside a small volume, making reactions much more efficient. If molecules were crammed together in a tiny bubble, they'll bump into each other way more often than if they were spread throughout the ocean. And it established a distinct internal environment that could be different from the outside world. This is essential for life. The formation of protocells was a crucial step toward true living systems, creating the first cells before life even existed. Now, as cool as the primordial soup theory is, it has some serious challenges. Scientists are still debating whether it's the right explanation. One major problem is the dilution problem. The ocean is huge. If organic molecules formed and washed into the ocean, they'd be spread out over an enormous volume of water, making the soup very, very dilute or watery. When molecules are super dilute, they rarely bump into each other, so reactions happen very slowly. It's hard to see how complex polymers could form efficiently in such a dilute environment. The molecules would be too spread out. That's why scientists think localized environments like tide pools or hydrothermal vents are important. They could concentrate the molecules. Another challenge involves polymerization thermodynamics. That's the energy rules governing how polymers form. Like we mentioned, forming polymers requires dehydration reactions which are difficult in water. Thermodynamics, the science of energy and entropy, says these reactions don't want to happen in watery environments. The polymers tend to break apart rather than form. Scientists are still figuring out how to solve this. There's also a problem with the early atmosphere. More recent research suggests Earth's early atmosphere was likely less reducing, had more carbon dioxide and nitrogen, less ammonia and formaldehyde than Miller and Urey assumed. In a less reducing atmosphere, it's harder to form organic molecules. Finally, there's the lack of direct geological evidence. We haven't found clear proof in ancient rocks that there was actually a vast organic rich soup in early oceans. One really interesting one is the chirality problem. Chirality means handedness. Some molecules can exist in two mirror image forms, like your left and right hands. They have the same parts but are arranged in mirror image ways. For amino acids, we call these forms L amino acids and D amino acids. L for left-handed, D for right-handed. Here's the weird thing. Life exclusively uses L amino acids to build proteins and D sugars to build DNA. But when you make amino acids through abiotic synthesis, like in the Miller-Urey experiment, you get a racemic mixture, an equal mix of both L and D forms. So why does life use only one form? How did early life select just the L forms? This is still a mystery. Another challenge is the harsh environment of early Earth. The intense UV radiation from the sun, remember, no ozone layer yet, could destroy organic molecules as fast as they formed. UV light has enough energy to break chemical bonds. This seems like a major problem. How could complex molecules survive long enough to evolve into life? Maybe life started in protected environments like deep underwater where UV couldn't penetrate. Finally, there's the problem of energy and metabolism. Modern life has complex metabolic pathways, series of chemical reactions that extract energy from food and use it to build things. It's hard to explain how these pathways developed from a passive soup. Some scientists think metabolism might have come first, driven by chemical reactions on mineral surfaces, and that the primordial soup idea is too passive. We've explored the primordial soup hypothesis from top to bottom. We started with early Earth conditions between 3.7 and 4 billion years ago, a reducing atmosphere rich in methane and ammonia, with lots of energy from volcanoes, lightning, and UV radiation. We saw how these energy could drive prebiotic synthesis of monomers like amino acids and nucleotides, and we learned about the amazing Mueller-Urey experiment that proved organic molecules can form spontaneously under early Earth conditions. We explored how Earth's early oceans acted as a giant chemical reactor, especially in concentrated environments like tide pools and hydrothermal vents. We discussed the tricky problem of linking monomers into polymers despite being surrounded by water, and we dove into the RNA world hypothesis that solves the chicken and egg problem by proposing that RNA was the first genetic material that could also act as enzymes. We saw how protocells could spontaneously form, providing the crucial compartmentalization needed for life. But we also confronted the significant challenges: the dilution problem, the thermodynamic difficulty of making polymers in water, questions about the early atmosphere's composition, mystery of homochirality, why life uses only one mirror form of molecules, the harsh UV environment, and the puzzle of how metabolism evolved. The truth is, we still don't know exactly how life began. Abiogenesis, the origin of life from non-life, is a vibrant field of active research. Scientists are exploring many ideas, including alternatives to the primordial soup like the hydrothermal vent theory, life starting at deep-sea vents, and new approaches like dynamic combinatorial chemistry, studying how complex molecular systems self-organize. What we do know is that somehow protocells evolved into the last universal common ancestor, the single organism from which all life on Earth descended. This transition likely spanned billions of years of chemical and biological evolution. Thanks so much for hanging out with me at Nog's house today. Next time, we're going to talk about one of the most important events in Earth's history, the oxygen revolution, when photosynthetic organisms started pumping oxygen into the atmosphere and completely transformed our planet. That's going to be awesome. I hope you'll join me for it. See you next time.